My name is Rong Ma. I'm a postdoctoral fellow at Emory University. I received the 2021 Michelson Prize for harnessing receptor mechanics as a marker for immunogenicity to isolate and identify potent T-cell receptors and recognized antigens. Curiosity has always been one of the biggest motivations that pushes me forward in research. My research focused on developing molecular sensors made of DNA, which can detect receptor forces on piconewton scale generated by cells when they are probing the surrounding environment. We and others in the field have found that the T-cell receptor employs mechanical forces as a part of its triggering mechanism in order to boost this specificity of antigen recognition. And by studying these forces, we found that the mechanical sampling of the antigen by the T cells is correlated with the T cell activation. So with this knowledge moving forward, we are trying to develop an assay to harness these mechanical forces transmitted through the receptors as a biomarker. This would be one of the very first attempts of harnessing the dynamic and biophysical nature of this antigen recognition process for potential cancer immunotherapy purposes. And in the long run, if this method is demonstrated to be effective for precisely identifying the potent T-cell receptor antigen bindings, we could use it as a strategy to generate personalized neoantigen cancer vaccine. As an early career researcher, the support from Michelson Prize will give me a lot of independence, as well as the peace of mind to focus solely on this research. And it allows me to pursue the high-risk, high-reward project of developing a novel assay for neoantigen and neoantigen-specific T-cell identification. Hello, my name is Rong Ma, and I am a postdoctoral fellow in the Salida Lab at Emory University. Today, the title of my talk is Mechanical Forces Help T-cell Receptor Distinguish Friend from Foe. T-cells constantly scan the surface of other cells to defend against infections and cancer. As you can see from this video, a cytotoxic T-cell is searching for antigens on an antigen-presenting cell. If we zoom in at this interface, we will find one molecular interaction, which is the binding between the TCR and the peptide C molecule. Once the TCR recognizes the peptide, it initiates a signaling cascade, and as an outcome, T cells get activated and produce these cytotoxic granules showing red and precisely release them to the target cell to kill. This process takes place at an interface, which has made us wonder, is there any physical cues during TCR recognition and T cell activation? For example, does T cells produce traction forces when searching for antigens? One particular question we are interested in is that are mechanical forces involved in TCR peptide C interactions, and do they matter? To answer this question, we decided to engineer chemically identical but mechanically different substrate to present peptide Cs for T cells. Our lab used tension gauge tether technology, which is a DNA duplex anchored on a substrate and present the ligand of peptide C to T cells. It would only denature when the pulling forces are exerted on this peptide C. These DNA duplexes can be mechanically melted at 12 piconewton in unzipping geometry or 56 piconewton at shearing geometry. And the difference comes from the positioning of the peptide C ligand for the TCRs to pull on. Just to give you an example of how tiny these forces are, 1 Newton is how it feels like to hold an apple. And the scale of the TCR forces are a trillionth of that. As you can see, when the force transmission from TCR to peptide C was terminated at 12 piconewtons, the level of the TCR triggering marker phosphorylated ZAP70 dramatically decreased compared to at 56 piconewtons. This data tells us that the mechanical forces are very important in T-cell activation. Now our question becomes, if the sustained mechanical forces is critical, can we visualize them? Our lab developed the molecular tension fluorescence microscopy, which uses DNA-based molecular tension probes to report mechanical forces exerted by the cells through the TCR to the antigen. These sensors are immobilized on a cover slip and report mechanical forces with fluorescent signals. When there is no force, the hairpin is folded and the fluorescence is quenched by the quencher. When TCR binds and pulls on the antigen on top of the sensor, the hairpin unfolds and generates a fluorescent signal. With this sensor, we observe TCR forces in real time. The green dots fluctuating under the footprint of these T cells are the forces exerted through individual TCR peptide C interactions. And by using hairpins that unfold at different forces, we got a very defined range of TCR forces between 4.7 piconewtons and 19 piconewtons. Since this sensor detects real-time TCR forces, we were curious whether the overall forces accumulated when T cells interact with peptide Cs would look different for peptides with different potency. So we introduced a complementary DNA strand to this system, which only hybridizes to the mechanically unfolded hairpins, and thus lock the tension signals for cumulative tension mapping and quantitative measurements. And as you can see here, N4 is the most potent antigen, which showed the strongest force signal. The other peptides, Q4, V4, and G4 only had a single amino acid mutation at the 4th position, showed a dramatic decrease in TCR forces despite the similar affinity. And here antibody is the control. We found that the TCR mechanics is highly correlated with the immunogenicity as opposed to the TCR peptide C affinity. Next, we ask the question whether the dynamics of the TCR forces are different. We further developed a method of extracting the force lifetime information from our tension measurements. Our result shows that the stimulating antibody, anti-CD3, has the longest force lifetime, and the cognate peptide N4 has an over 10 times longer lifetime than the less potent peptide Q4 for forces greater than 4.7 piconewtons. This result echoes with some single molecule force spectroscopy studies when they use a peptide C-coated bead to apply force through a single TCR on a T cell. It is noticed that a few TCR peptide Cs form catch bonds, of which the peak lifetime of the binding is at around 10 piconewtons. And many peptide Cs form slip bonds, of which the binding lifetime decreases as force is applied. It is not only reported in the mouse OT1 model that I've been using, but also in human TCRs. For example, Garcia lab at Stanford reported 2 TCRs both binding to the peptide C at the same affinity, but only cells with TCR 589 were activated. The differences among these 2 TCRs are that the TCR 589 forms a catch bond with the peptide C, and the non-activating TCR 55 forms a slip bond. With that, I'd like to conclude my talk. What we find is T cells generate 4.7 to 19 piconewton tension against peptide Cs, and terminating this force transmission at 12 piconewton dampens T cell activation. Moreover, The TCR mechanics is correlated with peptide potency, and the TCR peptide potency force lifetime at 10 piconewton is one key difference that decides activation or no activation. Hopefully, we and others' research on TCR mechanics would inspire new immunotherapy that has the force component integrated to it. At last, I want to say thanks for this opportunity to present some of our lab and my research. My acknowledgements go to my advisor Khalid and the Salaida Lab at Emory University, as well as our collaborators. I would like to thank our funding sources, NIH, as well as the recent support from the Michelson Foundation and the Human Vaccine Project. Thanks for listening to my talk, and I hope you find it interesting.